Describe the digestive role of chymotrypsin. Which two other digestive enzymes of the same category are secreted by its source gland? So students, here understand the question. It is little bit tricky. So first part says explain the role of the chymotrypsin. Next part is which two other digestive enzymes of the same category are secreted by its source gland. So what is a chymotrypsin? It is an enzyme which helps in the digestion of protein. So that means we need to know two other enzymes which helps in the digestion of the protein. So the question says which two other digestive enzymes of the same category. That means we should learn about the enzymes which helps in the digestion of protein and secreted by its source gland. So chymotrypsin is secreted by the pancreas. So we need to learn the other two digestive enzymes that digest proteins secreted by the pancreas. The pancreatic juice secretes three inactive enzymes, that is trypsinogen, chymotrypsinogen and procarboxypeptidase. So these are the three protein digesting enzymes produced in the pancreas and they produced in the inactive form. Now here in this question, let us learn about the functions of all the three enzymes. Now firstly, coming to the first part of the question, describe the digestive role of chymotrypsin. So chymotrypsin is the active form. Its inactive form is chymotrypsinogen. So this inactive chymotrypsinogen is converted to its active form by the enzyme trypsin. So trypsin once gets activated, it actives the other inactive enzymes such as chymotrypsinogen and carboxypeptidase. Now once the chymotrypsin is active, it acts upon the protein and it breaks the protein into peptides. It partially hydrolyzes the proteins into, protein is a chain of amino acid. So the chymotrypsin breaks this chain and it becomes peptides. Peptides are nothing but smaller chain of amino acid. So the role of chymotrypsin is to break the proteins into smaller peptides. So here we can see the chymotrypsin acts upon the proteins and breaks them into peptides. So peptides are nothing but a small length of amino acids. Coming to the second part of the question, which two other digestive enzymes of the same categories are secreted by its source gland? So the same categories are trypsinogen and carboxypeptidase. So trypsinogen is the inactive form of trypsin and procarboxypeptidase is the inactive form of carboxypeptidase. Now, once the food enters into the small intestine, the mucosa, that is the inner wall of the small intestine, releases an enzyme called enterokinase. Now, this enterokinase acts upon trypsinogen and converts it into its active form, trypsin. Now, once the trypsin gets activated, it breaks the proteins into peptides. So, the activated trypsin hydrolyzes, means it breaks the protein and converts it into peptides. Now, similarly, the activated trypsin, here we can see, once the trypsin gets activated by enterokinase, it activates the other two enzymes, chymotrypsinogen, which we learned in the first part of the question, and procarboxypeptidase into carboxypeptidase. Once the carboxypeptidase gets activated, it acts upon the carboxyl end of the peptide. So every protein has got two ends. One is the amino end, the other one is the carboxyl end. If this is the peptide, and here if it is the amino terminal, and here if it is the carboxyl terminal, the carboxypeptidase identifies the carboxyl end and starts acting upon the amino acid present near the carboxyl end. So it breaks this peptide bond and releases the amino acid. So the peptide will become shorter in one amino acid. So a small peptide chain and one amino acid from the carboxyl terminal will be released. And again it will break the other amino acid and this process goes on. So we learned about the chymotrypsin. So chymotrypsin breaks proteins into peptides. So trypsin also breaks proteins into peptides, whereas carboxypeptidase, it removes the amino acid from the carboxyl end, and all the three are released by the pancreas.